This is the plan of the lecture. We're going to go over the way that people do ChIP-seq experiments and some of the technical details that are again useful for interpretation of ChIP-seq data. We'll go over some quality control aspects of ChIP-seq data. We'll talk about peak detection for ChIP-seq. And we'll talk about peak analysis, interpretation, basically peak annotation. And if we have time, we'll talk about, we'll present a few interesting ChIP-seq papers. I do want to say that the lab for this will be mostly tomorrow. Today we're going to do about 2 hours and 30 minutes of lectures. We'll have a little break. Matt will also give you a small lab, 30 minutes lab. The main goal of that lab for today will be to teach you how to build read count profiles from a BAM file, something that some of you guys asked about in the past few days. So that'll be the lab for today, just 30 minutes or 25 minutes. And tomorrow we'll talk about peak detection in ChIP-seq. We'll actually show you how you can do it in R. We'll teach you how to do peak annotation, ChIP-seq peak annotation. We'll teach you how to do de novo motif analysis of ChIP-seq peaks, basically using R. So that'll be tomorrow. So today I'm going to tell you about a few things. So the first thing is ChIP-seq experiment design, basically. And how, again, people do ChIP-seq in the lab. So we had a quick introduction about ChIP-seq a couple of days ago. I do want to give you a few more details. So ChIP-seq is, the idea behind ChIP-seq is that you have an antibody that recognizes a protein of interest, a transcription factor, typically a protein that binds to the DNA. And you want to know where the protein binds on the genome. For example, on the human genome. So what you basically do is to pull down the protein together with the DNA that the protein binds to, right? And you take the DNA and you sequence it and you map it back to the genome and it tells you where the protein binds. The way that the experiment is done is like this. You start with cells. You cross-link the cells with formaldehyde. And the cross-link, what it does is basically it strengthens hydrogen bonds between molecules. So whenever you have a hydrogen bond, formaldehyde is going to convert this bond into a covalent bond, a very strong bond. It solidifies all of the otherwise weak interactions between molecules. Okay, and so you want to solidify the interaction between the DNA and the proteins that bind to it. Okay, that's what cross-linking is doing. Once you've done the cross-linking, you isolate DNA from your cells, right? And the next step is to chop your DNA into small pieces, and you do that using a process called sonication. And essentially, you use ultrasound as a way to damage and destroy the DNA, cut it into pieces, essentially. You add an antibody that's going to recognize a protein of interest, right? So it's going to bind to the proteins like this. This is the antibody here. It binds to the proteins, right? And using different techniques, but for example, beads, you can essentially pull down specifically the protein DNA complexes using the antibodies. Okay, so it allows you to isolate, to purify the complexes between protein and DNA, right? And the next step is to take this DNA to release the cross-links using a chemical approach. That basically leaves you with protein and DNA. You get rid of the proteins, that leaves you with DNA. And you take this DNA and you sequence it using high-throughput sequencing. Okay, that's basically the idea behind ChIP-seq, right? Yeah.
Question. I wonder, can you do ChIP-seq in conjunction with RiboSeq that we heard about the other day? Like, imagine you know that, or you think that some protein is in complex with the ribosome, but you don't really know, and so you want to pull down basically the protein as it's attached to the ribosome, and then see, okay, is the protein binding to the ribosome at particular points of the genome or something like that?
Yes. I mean, it wouldn't necessarily. I mean, it would be very different from ChIP-seq in the sense that there will be no DNA involved. I mean, the ribosomes are not, the nucleus are outside. So, what you could do and what people do is to isolate, polyribosomes and the mRNA that's bound to the ribosomes, and take this mRNA and then sequence it, right? So it's similar to what you're talking about, except that usually it doesn't involve antibodies. The ribosomes are actually reasonably easy to isolate. They're just very big machines, and you don't have to use antibodies.
So for a particular protein that's bound to, like if you're interested in a particular protein that's bound to the ribosome complex, you would then, I mean, could you use an antibody to pull down?
Well, antibodies are used for many things, including as a way to determine whether things co-localize, whether things are forming a complex. So if you have an antibody against ribosomal component and an antibody against another protein, you can actually do co-immunoprecipitation and determine whether essentially when you pull down both proteins at the same time in the same complex. So you can definitely extend some of these ideas, and in fact some of these ideas actually predated ChIP-seq. ChIP-seq is a recent technique. This pull down, this co-immunoprecipitation. This is something that predated ChIP-seq by a long time. So this is ChIP-seq. Every time that you do ChIP-seq, you want to do a control experiment, which is pretty much the same as a ChIP-seq experiment, except that you basically forget, you omit to use the antibody, to add the antibody to the mix. What you end up with is essentially isolating DNA, but not DNA that has been selected for binding to the protein of interest. Okay. Anybody knows why it's important to do this this control here? What you get signal? What kind of signal do you get? Yeah. So what makes you think that the idea here is to basically just get random DNA, right? So one could think that you wouldn't need to do this experiment because random DNA could be seen as sort of uniformly distributed across the genome. Is there a very specific reason why you want to do this experiment here? Yeah.
So there is some sequences that get over-amplified through like during Illumina, for example, when they process. So then you can see, if you see the same piece in the chip and in the input, it means that this is just not the enrichment coming from the antibody, but just because this is the property of that sequence.
That's right. So basically, this is one reason, but set of reasons why you want to do this experiment is that some of these steps here, including the sequencing, like you say, do involve actually some enrichment. And it's not only the sequencing, it's also isolation of DNA and sonication. So when you sonicate DNA, right, which is, again, is breaking the DNA into small pieces, right, the sonication is not equally active or efficient across the entire genome. If you have chromatin, it's very tight, right, heterochromatin. Sonication has a hard time breaking the DNA in these chromatin regions. If a chromatin is open, if the DNA is kind of naked, sonication is very good at chopping the DNA into locations, right? So what's happening, in fact, when you just isolate DNA out of this process, sonication, and cross-link and sonication, you actually get more DNA in regions where you have where the DNA is open, right? In fact, there are actually people using this feature, this sonication preference, or this bias sonication efficiency, to actually locate regions of a genome where the DNA is open. There's a technique that's called sono-seq, which is basically using sonication plus sequencing as a way to identify regions of the genome where the DNA is open. Okay, and that allows you to identify regions like promoters, regulatory regions, where the DNA is usually open. It's not, a particularly good technique. There are better techniques to identify these regulatory regions, but this is, something that people have published. So just to illustrate the fact that there are biases that are created by the sonication, and again, additional biases that potentially are created by the sequencing. Okay. So this is what people do when they do ChIP-seq. So the first step when you do ChIP-seq is to identify an antibody that's ChIP grade and that has high specificity. Okay. And there are multiple ways to do this. Typically Western blots or peptide arrays if you do ChIP-seq for histone modifications. This is a very important step here. This is a very important step because there is a lot of variability among antibodies. There are batches of antibodies that are good, batches that are bad. The same antibodies sometimes can actually vary in terms of efficiency. This is an illustration of this here, actually. So this is something we've done in collaboration with some people at Cornell, where we looked at the same antibody. That was an antibody that would recognize H3K9 translation. It's a histone modification. We looked at two batches of the same antibody from the same company, right? We looked at what kind of histone modification this antibody actually recognizes, right? It's sold as a K9 trimethylation antibody, right? So you can use these peptide arrays where each dot is a different peptide with a different histone modification, right? And you can basically tell where your antibody is binding within this array, right? As you can see, so each sort of line here corresponds to a different histone modification. As you can see, so these are the K9 translation peptides, and these are some of the other peptides on the array. In this particular batch here, the antibody is only binding to the K9 translation dots, wells if you want, right? But another batch of the same antibody, again from the same company, if you look here, right, you see that this antibody here, this batch of antibodies is actually recognizing a whole bunch of additional histone modifications. Okay, so it's recognizing multiple histone modifications, not only K9 translation. Okay, what that means is that if you were to use this antibody here to pull down DNA as a way to identify where you have this histone modification in the genome, you would not only pull down DNA associated with these marks, you would pull down DNA associated with also these histone marks. Yeah.
What's in the columns?
The columns are just different peptides. Essentially, you have a peptide. There's typically a lysine somewhere in the peptide, but the peptide has different length, and the lysine is not always right at the same location. Okay.
So,
yeah.
Yeah, this is really interesting, but I would suspect that most people who publish on these anyways don't do this, so that's probably...
Now people do this, but before several papers were published about this problem, this paper is actually very recent. It's very recently that you recognize that two batches of the same antibody actually can be very different in terms of cross-reactivity and efficiency. And in fact, there are lots of papers that probably actually didn't do this type of analysis, and probably have reported wrong results due to these problems.
So are these now available?
Yeah, these you can actually buy now. These peptide arrays, absolutely. Yeah. But this is a very nice technology now that's available commercially that used to not be as available basically.
So this is the same antibody from the same company, just different batches?
Yeah, that's right.
If you encounter a situation like this, would it be to get another batch? Is it to switch antibodies? Is it to switch, what's the...
Absolutely get another batch, and definitely get a refund for this. Because it's not performing as advertised for sure. Yeah, that's the only way. Definitely get a different batch. Okay. So once you have an antibody that works well, this it's specific, and it's not cross-reactive, it doesn't recognize everything. Then the next thing to do is to optimize conditions for the pull down for the IP. Typically, you do that with single locus ChIP-PCR, where you have primers and amplifiers a region of interest. You have positive controls, negative controls, and you try to maximize enrichment in the positive controls and minimize enrichment in the negative controls. That's basically something that you have to do, every time that you start a new ChIP-seq experiment. And then you sequence your ChIP product. This is a semi-old slide in a sense. You know, it used to be that you had to sequence only a full lane, but now you have Illumina kits that allow you to multiplex and sequence multiple samples per lane. We typically do single read. There's no good reason to do paired end ChIP-seq. It doesn't really help you, you know, with in terms of peak detection or you know, all the tasks. And then you sequence input or IgG as a control. IgG is just a control antibody that's not supposed to recognize anything interesting. So this is just an illustration of what you actually do when you pull down this DNA fragment, so what you actually get when you pull down this DNA fragment. This is meant to represent the protein here, and this is meant to represent the DNA fragment that's a double-strand DNA that you pull down with the protein itself, cross-links, you know, being here. The goal of this picture here is just to show you that the DNA fragments that you pull down are actually much larger than the size of the actual protein-DNA interface, right? A protein-DNA interface, which is something that Harman discussed this morning, is actually a very small segment of DNA, right? The technology, the sonication, you know, the amount of sonication that you do essentially gives rise to much larger fragments. This is an illustration here of the sonication process, and it's just to show you that the more you sonicate, the longer you sonicate, the smaller and smaller fragments you get. So this is DNA fragment size here. This is sonication time. And it shows you that you sonicate for 20 minutes. You get smaller and smaller fragments. And typically, the kind of fragments that you know, sequencers you like, you know, to sequence. This is going back to yes.
By soncating much more, is there any chance that you actually start to unbind?
Yeah, so the idea is that you do want to sonicate a lot so that, you know, the fragments are smaller and smaller and you get closer and closer to the protein-DNA interface. But if you sonicate too much, you actually start breaking the bonds between the DNA and the proteins. And therefore you decrease your pull-down efficiency. So there's definitely a trade-off. You know, you can't sonicate forever hoping that you're gonna, you know, end up cutting, you know, here. There are other techniques that one can use to narrow down the piece of DNA more to the protein-DNA interface here. One of these techniques is called ChIP-exo, where you actually use an enzyme to digest the DNA that's not involved in cross-links with the protein. It's good, but it also means that you have to start with, you know, with a very large amount of DNA because the enzymes is, you know, just damaging the DNA and reducing the amount that you get in the end. Yeah, so this is, you know, there's an average length or a length of DNA fragment that's good for sequencing. But what I also want to illustrate is the fact that, as you know, with Illumina sequencing, we're not sequencing entire fragments. It's actually doable to sequence with longer reads like this. But again, there's no real reason to do this, no good reason to do this. And what you typically do is you sequence one extremity of a DNA fragment, right? Again, if you were to do paired end, you would sequence both extremities, but again, there's no good reason to do this for ChIP-seq. So you sequence one extremity. Typically, the extremity that you sequence is chosen at random. It could be one this side or the other side. And the size of the piece of DNA that you sequence is somewhere between 40 and 100 base pair. Okay. I mean, all that's important in terms of analysis, as you'll see in the next few slides. Okay. Right. So this is going back to the alignment process itself, which is often done using a program called BWA. We went over BWA last time. This is exactly the same process here. This is again, you know, downloading the reference genome, indexing the genome, aligning the ChIP reads to the genome using the same commands that I showed you last time, and then also aligning the input reads to the same genome. Essentially, you end up with two files, and you're gonna compare these two files for peak column.
Can I ask a question?
Yeah.
You talked about advantages between bow tie and BWA, like variant
Yeah, so so the key aspect about analysis or alignment of reads in ChIP-seq is that you're usually not interested in variance, right? You just want to know where things map in the genome, and then you want to count how many reads map to a given location. So the ability, of these aligners to detect, mutations or variations is not as important. That being said, typically your reads are coming from a given sample, coming from cells, that have a genome that's different from the reference genome that you're going to use for mapping. And if your aligner is good at allowing mismatches, just polymorphisms, that exist between genomes, then you might be able to get more reads as a consequence of using a better aligner. And the fact is that if you use BWA, and again, that's these efficiencies are changing because programs are also improving all the time. But BWA used to be slightly better at allowing indels in your reads, essentially taking into account short insertions or short deletions in the reads. And when Bowtie wasn't doing that well, you would see that BWA would align somewhere between 2 and 5% more reads than Bowtie because of its ability to identify indels. That does not seem to be the case anymore. They are about equal now, and Bowtie is actually a bit faster. So we've just been using BWA for a long time, which is why I have this slides. But Bowtie and BWA are equally good for this, for ChIP-seq. Yeah. Okay. This slide here is just meant to show you that, like we discussed last time, sometimes your read map to the reference genome to a unique place. Sometimes your read map to multiple locations, right? And we discussed, what one can do about this last time. A solution is to remove all of our reads that map to multiple locations. Another solution is to pick a random location. Another solution was to duplicate a read, if you remember. All of these solutions, usually when you have a read that maps to, let's say more than, 20 places, usually you take the read out because you really have no idea whatsoever where it's coming from. But all of these approaches to deal with multi-mappers apply to ChIP-seq. This is a slide to illustrate that reads can map in both directions on the genome. The ChIP-seq, again, remember the way that the extremity of a DNA, which extremity DNA is chosen to sequence is random. What that means, given that the reference genome is stranded, is it's only one direction if you want. It means that your reads are going to align equally well on both directions, right? On both strands. There's a convention about representation, especially in tools like the genome browser, where reads that go in one direction are blue and reads that go in the other directions are red. I don't remember which one it is, but there's basically blue and red reads. We'll see more of this in a few slides. This is the SAM format again. This is typically what you get out of the aligners. We'll discuss this. So no point in discussing this further. Again, yeah, you don't really care about variance. You don't care about differences between sequence differences between the reads and the reference genome. That said, actually, we did publish a paper last year showing that you can actually identify mutations from ChIP-seq. We actually used the reads. We actually looked at the reads. It was in the context of pulling down a particular type of histone modifications that's prone to mutations in B cells. In B cells, there are these enzymes that mutate the DNA and try to make better antibodies. And basically, using ChIP-seq, we showed that these enzymes are actually binding to actually a lot of promoters and making point mutations onto these promoters. It was a kind of an exotic use of ChIP-seq, and typically you don't look at mutations in the reads. You care mostly about counting how many reads map to the genome. So this is some of the procedures that you would do for quality control for ChIP-seq. The first, a very important one is to estimate how many clonal reads you have in the genome. Can anybody remind me what clonal reads are? What's a clonal read? What do you mean from the same cluster? So like a read that's duplicated from the same cluster? That's not, the cluster growth is definitely producing multiple copies of the same DNA fragment, the same location in the slide. But that should not end up in your sequencing data because the software is reading the entire cluster, the fluorescence from the entire cluster. This is actually, these are reads, clonal reads or reads that are in the sequencing data that you have to deal with.
Sorry, identical reads, right? So you don't know if it's a PCR artifact.
That's right, that's right. So basically these are reads that are identical in sequence, mapped to the same location and the same orientation, right? And these are usually PCR duplicates. These are usually a result of PCR duplicates, usually a result of people not having enough DNA to start with to do ChIP-seq, to essentially to not enough DNA to put into the sequencer. So they have to do multiple rounds of PCR amplification so as to get more DNA. But because the PCR amplification has biases, it amplifies certain fragments better than others, you end up with multiple copies of the same DNA fragment, and you have to deal with this. Yes.
Did you know that there's more clonal reads in the ChIP-seq than RNA-seq data, or?
It's about, I would say it's probably about the same by experience. Actually in my experience, there are probably more clonal reads in RNA-seq compared to ChIP-seq. And one of the reasons is, we talked about this last time, is, there are some genes that are highly expressed, and that do contribute reads that are kind of clonal looking. They might not be clonal, just, the RNA is super highly expressed and you sample multiple times the same read. So I think there are usually more clonal reads in RNA-seq. But there is a way to check that in your sample. What you can do is to count the number of reads, the number of tags. These are figures from a website that uses the word tag instead of reads, but it's the same thing, right? This is a figure that shows you the number of tags per genomic position. So across the genome, you go nucleotide by nucleotide, and for each nucleotide, you count how many reads map at that location, and you should see something like this, where you have very few locations where you have more than one read that maps to the same location, same orientation. That means that you have a low clonality rate for your experiment. If you have this, right, where you have many positions that have a significant number of duplicate reads, it tells you that there's a huge amount of clonality in your sample. So what do you do when you have, what do you do with clonal reads? What would you guys do with clonal reads? You collapse them, that's right. You collapse them into one. Yeah, that's what people do. What that means is that it actually effectively decreases the number of reads in your sample. But frequently with ChIP-seq, you're left with enough reads, unless your clonal fraction is like 90%, you're left usually with enough reads to do peak calling. But the fractional clonal reads, I mean, again, it's an artifact from the PCR. It does not mean that if you have a high level of clonal reads, doesn't mean that your sample or your experiment has failed. It means that you have to collapse them and take into account this clonality. It doesn't mean that your experiment has failed. You should look after collapse if you have, using genome browser, for example, if you have, still have sharp peaks. It may just be that, you did too much, PCR amplification, but again, the collapsing takes care of this PCR amplification and gives you the signal. So it's not necessarily a problem unless you have, again, 95% or 99% clonal reads, which we've seen. Yeah.
What exactly are genomic positions here?
Genomic position is nucleotide in the genome.
Across the whole genome?
Across the whole genome, that's right. Yeah.
So mostly you would expect to be zero.
Yeah, so zero is not shown, but yeah, absolutely. Yeah. You would expect to see a huge bar zero. Yeah. Comment.
So I could imagine two things that drive this. On the one hand, if the number of molecules that you start from as your sample, right, if it's comparable to the number of reads, then you have a problem and you kind of undersample it. Or it could be biases because of the PCR, right? And
that's right.
somehow you push some sequences more than others. Is it clear what drives this problem?
Oh, it's pretty much only the PCR amplification that drives the problem. Yeah, for sure. Yeah.
The biases in the amplification.
Yeah, no, and you can tell, when you look at input DNA, which is actually, this is no, I thought this was an input, but if you look at input DNA, which is unbiased, random DNA, I mean, you don't get usually this, or you get less of this problem. Yeah. It's yeah. I mean, yeah. It's.
That said, the PCR bias is most pronounced when you start with very small amounts.
Yeah. But it's really because you have to amplify. That's really the. When you start with small amounts, the natural thing to do is to do many rounds of PCR to get to the amount that you need for sequencing, and therefore that leads to this problem. Yeah. So I do want to mention that so these are analyses that you can do in R, but there are also tools like Homer that actually do a pretty nice job producing these figures for you. It's not an R package, but it's a very nice set of tools that you can use to create some of these plots. But the other quality control procedure that people do is a fragment size analysis. This is based on what a peak looks like in ChIP-seq. So this is what a peak looks like in ChIP-seq. As you can see, there are lots of these blue reads on one side of a peak and lots of these red reads on the other side of a peak, right? And that is basically, again, this is basically due to the fact that you sequence either extremity of the same DNA fragment, right? What that allows you to do is to estimate the size of the DNA fragments that you sequence in the first place. We'll get back potentially to this figure, but this is another figure showing a nice peak, and again, this blue and red, some double distributions with potentially a small peak here. That another story. Yeah.
Compared to the sort of random jumps that you get, do these tend to be more equally distributed between two strands, or is the random jump also equally distributed?
So are you talking about whether there's a bias between the strands, or there should be about 50% reads coming from one strand, 50% from the other strand?
No, what I mean is where there's a fake peak, does it have the same pattern where they're accumulated sort of in a bimodal distribution, or so that you can use that to recognize these things?
Yeah, no, so that's actually what people do. People do use that actually feature to recognize peaks. It can be seen as a feature of peaks. There are complications, and I'll show you more about this, and this is why a lot of people, including us, actually do not use this feature. The complication is due to when you have two peaks that are very close to each other, and kind of mess up, the nice, red on one side, blue on the other side distribution of reads. But this is a feature that's actually used. This is something that's used, for identification of peaks. It just, works better when you have an isolated peak like this. It probably would work okay in this situation, but if this peak here was closer to this other one, the distribution would be kind of mixed up, and the approaches that use these two shifted distributions would break down. But the presence genome-wide of this shift between the two distributions is an opportunity to do some quality control. And one of the quality control is to basically use this feature as a way to get a sense whether your data has peaks. Your data is kind of peaky, if you want. And one way to do this is to look at tag autocorrelation, distance autocorrelation. So the way you go about this is that you calculate the distance between reads in one direction, say blue reads, right, and the next read in the same direction, okay? And if you were to do that genome-wide, you would actually get a distribution like this, right? And what you can do is to do the other analysis, which is to look at read in one direction, blue reads, and look at what's the closest read in the other direction, right? The closest red read like this, right? And if you were to plot the distances of the distribution of these distances, you would see that this distance, the second distance, is actually like this. And the shift between the two distributions actually gives you an information about the length of the DNA fragments. Okay. Again, it goes back to the picture that I showed you about, the protein, the reads that you sequence from the DNA that's pulled out of the protein. The point being that in a ChIP-seq experiment that has peaks, structures, this distance here should be reasonably large and should correspond to about the DNA fragment size that you put into the sequencer, if the experiment worked well. Again, the intuition is, think about this. Let me see if yeah, this is probably better. Think about a blue read here, right, and think about what's happening next to it. Think about if you have a blue read going in that direction, the distance to surrounding red reads is going to essentially you're going to have a distribution that's going to be high around that location, right? So that if you do that genome-wide for all of the peaks and all of the reads, you get a distribution like this, and again that allows you to estimate the distance between the fragment size. Now this fragment, as I was telling you, has to be close to what you saw in the gel, after sonication. And if you have an experiment where you don't have a good peak content, if your data set is not peaky, doesn't have too many peaks, if basically the DNA fragments, the DNA reads are kind of mapped randomly across the genome, you would get actually more of a distribution of distances like this, right? That show very little difference. Basically, the difference between these two, the two modes of its distribution is equal to the size of the reads, right? That is a failed experiment that doesn't have any peaks. It's, the enrichment is not present in this. It's not a peaky data set, okay? So this is a nice way to determine genome-wide whether you have an experiment that worked and that gave you nice peaks that you can pursue in terms of analysis, okay?
So if it's a good experiment, you just see more separation.
That's right. If it's good, yeah, you should have more separation. That's right. So the separation should be about the size of the DNA fragments that you sequence, of the average size of the DNA fragments that you sequence, as you can see here. Right.
So is this distribution driven by the clonal?
No. This is purely the peakiness of the data set. Again, it kind of goes back to this, where if you look at the red, blue read here, if you look at the distance of all of the surrounding, from this blue read to the surrounding red reads, you're going to have a peak, that's located about, the fragment size and length, to the.
That part I understand, but I'm confused why you would get this typical read length offset with the reverse trend, right?
It's the other strand. You look at the other strand. So you look at blue. Oh, you mean in the same strand, or?
But that other read, unless you compare them, or the other read could come from another IP fragment.
Well, this is
clone
also due to the peakiness of the data set. I mean,
this one I understand, but the next slide that you showed with the
this one here.
field experiment.
Oh yeah, this is just if your reads are randomly distributed across the genome with no peaks at all. So you don't have this closeness of this sort of increase or enrichment in terms of closed.
Right. Why would you get any correlation with reads mapping to the reverse trend? They'll be independent of each other.
You mean why do you even see?
Why is the red curve even peaked at 30 something or whatever?
Yeah, I need to think about it a bit more.
If it's clonal, you could eventually get double-stranded duplicates, right? And then you could either sequence it forward or reverse strand in that.
I see what you mean.
So it could be that this is totally driven.
You might be right. I think that's what it is. Actually, the reason for this is actually that you have clonal reads. Yeah. It's possible. Yeah. The difference is the size of the reads. Yeah, absolutely. Yeah, the shift between the two distributions is the size of the reads. I think you're right. It's due to the clonality of a sample. Yeah. I need to think about it a bit more because clonality is usually defined. It depends on how they define clonality in this, but clonality is usually defined as the same position and same orientation. So, yeah.
Yeah. Isn't the blue line intra-strand and the red?
Yeah, blue line is intra-strand. Yeah, absolutely. Yeah. Yeah, intra within the same. It's basically the distance between a blue read and the next blue read, and the next thing.
So the red line is inter-strand.
That's right. Yeah.
So that would be why you get the
Well, not clear that there should be a. Oh, you know what? I know what it is. It's due to where you look. You know which direction, which side of a read you look at. Whether you look at you know the 3' or the 5' of a read.
Oh, okay.
That's what it is. Yeah. Because you would actually count a read coming this way by looking at the 3' location basically on the other side. Yeah, so basically this is what this is a kind of a random peak-less data set here. Another quality control measure that you can look at is GC content. This is more about the quality of the sequencing itself. If you have a sequencing quality that's good and that didn't introduce any bias in terms of which fragments get sequenced better than others, you would expect to see some kind of reasonably straight line in terms of GC content or nucleotide content across the entire length of a reader, except at the beginning where there are usually problems. Sometimes the index, is kind of contributing. The adapters are contributing a couple of nucleotides to the beginning. That's why you have this bias here, but the line should be reasonably stable across the entire read. Again, this is detecting potentially sequencing issues. You see this is a weird experiment here where you have a change, you have a bias in terms of nucleotides at the beginning of a read, kind of goes away towards the end. This is a similar analysis comparing the amount, the content, the GC content of a reads from your sample to the GC content of a genome. Basically, you should not see a shift or a very significant shift between the genome that you're sequencing and the reads that come out of your sequencer. If you see a shift, it means that the sequencer is biased against certain fragments, or maybe the PCR also introduced the bias and so on. Okay. But again, very important quality measures that by and large you can do in R. Some of them might be a little bit harder to implement in R than others, but this is actually pretty easy to implement in R. You can calculate GC content of all your reads and build a distribution like this reasonably easily. Okay. Other quality control measures that people use, number of peaks. It's a very simple quality control measure, but it's related to the peakiness of a data set. But the bottom line is that if you have a ChIP-seq data set and you call peaks and you get 0 peaks, it's usually not a good sign. It tells you that your experiment probably didn't work very well. For most transcription factors, you do expect very sharp peaks in the genome. For histone modifications, the story can be a bit more complex. You have histone modifications from domains with not a huge amount of enrichment, but even so, they should usually form peaks, especially at promoters. Motif enrichment is also very, very, yes?
Can I ask you how many peaks are few?
Yeah, so, if you get less than 200 peaks, this is usually not a good sign.
What's a typical number?
Yeah, for a transcription factor, the factors bind to a lot of places. I mean, part of it is because the genome is very large, but transcription factors bind to many, many locations. Not saying anything about the functionality of this binding, but it is a fact that factors, most transcription factors bind to several thousand locations. So if you get less than a couple of hundred peaks, I mean, it's usually not a good sign. Yeah.
So if you have replicates of ChIP-seqs, you look at them individually, or?
Right. So that's a very good point. So and this is very important, obviously, as a question. There used to be a time 2-3 years ago where people felt that they didn't need to do replicates when they do ChIP-seq experiments or were doing RNA-seq. And then they sent out their papers to review, and those papers got killed pretty much systematically by reviewers because it's a very easy criticism. So then people went back to do replicates for all these RNA-seq and ChIP-seq experiments. And the question becomes, how do you combine these two replicates, for example? There are multiple ways to do this. And, not to say that there is a good way to do this, but there are multiple ways to do this. One is to identify what's your best experiment. You have two replicates. What's your best experiment, right? That's the one that you're going to use for the paper. What you can do is to call the peaks in this very good experiment, calculate the height in this good experiment. Now you take the same locations, the same location of the peaks. You go to the other experiment, and you calculate the height of the peaks in this other experiment. And you compare the height of the peaks between the two experiments, right? You can build a 2D plot comparing peak heights at the same locations, right? And if your replication was good, you should have a very strong correlation between peak heights at the same location between two experiments. This is what we've done in the past, among the things that we've done in the past as a way to compare ChIP-seq experiments. So as I say, you can decide to take only one, the one that works best, and use that for the next analysis. What you can also do after showing replication of the peaks at the same location, you can actually merge the two data sets, right? You merge the reads, right? You recall peaks, right? And that's what you use for analysis. What you can do is also to take the union or the intersection between the peak locations, right? You can say, okay, I'm only going to select the strong peaks and the ones that come up in the two experiments. Peaks that come up in one experiment, I'm not gonna look at them in my analysis. So they're all different strategies, and again, none of them is clearly better than the other one. It kind of depends on how stringent you want to be, how many peaks you want to rescue, how many peaks you want to end up in your analysis. Okay. Yeah, so I did want to add something about motif enrichment. When you do a ChIP-seq experiment for a transcription factor, right? Usually, by now, somebody would have done a similar experiment, would have studied a transcription factor or a transcription factor of the same family, and people would have a good idea of what kind of DNA recognition site this factor or this family of factors recognizes in the genome, right? So if you do a ChIP-seq experiment, you call peaks, you do motif analysis, de novo motif analysis, and you don't get any motifs for anything that looks like the factor that you're doing the experiments for, it's also not necessarily a good sign. It tells you that potentially your experiment didn't work as well as it should have, right? This is about the expected motif enrichment in peaks. Okay. We'll get back to this hopefully in a few slides. So let's talk about ChIP-seq peak calling. There are multiple tools to call peaks from ChIP-seq data. This is probably one of the first ChIP-seq peak caller that was created. It's pretty good. It's a pretty good tool that a lot of people use. It's not to say that it's perfect, but it's it's used by a lot of people. It's based on this idea. The idea is reasonably simple. What Max is doing is to first estimate from the ChIP-seq data in very much the same way as what I showed you for quality control. It's going to estimate what's the sort of average peak width in your data set. What's the peak width in your ChIP-seq data set? It's going to estimate this using the two curves that I showed you about. These two curves that are based on distance between red reads and blue reads. That gives you an average size of your peaks. And then what Max is doing is going back to the reads using knowledge of his size. So this is done using peaks that are very clearly peaks. There are always peaks in the data set that are super, super sharp, right? And that allow you to estimate this distance. Max is using these very, very clear peaks and basically reanalyzing the data for these peaks that are a little bit less clear using this approach. So it's taking this window here that was estimated from the data from these very, very strong peaks and basically counting how many reads map in a window which is twice the size of this thing here. And basically it's counting how many reads map in this window in one strand, how many reads map in the other window in the other strand. It goes back to what we discussed about using again, the shift between the blue reads and the red reads as a way to detect peaks. Yes. Yeah.
So how do you define a high quality peak? Is one for
So very, very sharp peaks. So it's a good, so I think they use a very simple heuristic as a way to detect these very sharp peaks. It's just sliding a window, counting how many reads, and looking at how many reads are on the side, and then but it's just they only need, I think, a few hundred peaks. I mean, for this, so it's really finding the very easy to detect peaks, and then reanalyzing the rest of the data using a more sensitive technique.
And is the input used in?
The input is actually used here. So this is the next step here. So this is using the Poisson distribution as a way to estimate or analyze the extent to which seeing that many reads in a given window of this particular size is surprising, right? Given the information that's given to you by the input data. So the input data again is this sort of randomly distributed genes across the genome. Again, not completely randomly. Hence the use of multiple averages for the background in this formula. But the bottom line, this lambda parameter here is coming from the input, and it's basically calculating the lambda values are calculated as the expected number of reads in the input in a given window. So here this would be the entire genome. So it's a window of the entire genome if you want. And then you have windows that are more local, different sizes. It's essentially counting how many reads in the input mapped to these windows divided by the size of the windows. That gives you an estimate of the expected number of reads per KB or in a given window. And basically, it's putting this into the Poisson distribution, which I think that you're familiar with by now, and it's basically counting, telling you the extent to which the number of reads that you see in the windows is surprising or is greater than expected by chance, given this expectation given to you by the input, right? And you can really do this type of calculation, in R. I mean, using the Depois function. I think Harman, you've told them about this, right?
It's good to connect because we talked about it this morning. Sort of binomial, the K expected is P times N, right? It's just the success rate times the size of the sample. That's what lambda is here. It's just a limiting case of the Poisson distribution. And then what we call K in binomial, that's X here, right? That's like the number of successes. And then lambda is the expected number of successes.
Right. And again, it's a p-value, which is the probability of observing a count that's equal or greater than the count that you observe. So this is the definition of a p-value, and this is what you get for each peak. So you get a p-value for each peak, or for each candidate peak, and Max is selecting the most likely, the best candidates, doing correction for multiple testing and so on, so that you correct for the fact that you're scanning very hard, the entire genome. That's basically what Max is doing. Yeah, and this is this is the peak caller that you're going to use tomorrow in the lab. That Matt is going to teach you how to use. It's a different way to call peaks, using a hidden Markov model. It's a bit more complicated, but basically what it does is it's a hidden Markov model formulation. So basically you have hidden states, which are the presence of a peak or not, and you have observed states, which is the number of counts, of read counts per window. And you have basically a model that's also using the Poisson distribution as a way to estimate whether in each window of a genome, you're more likely to be in a peak state or in a non-peak state. This is basically using a hidden Markov model. Yeah.
Both of these rely on having one thing right in the middle between your red reads and your blue reads. So if you're looking at a trimethyl P27 mark or something that's spread out over a large distance,
Yeah, those methods are not easily applicable, I mean, to this chromatin modifications that do not form peaks. You're right, it's absolutely based on, this visual, shift, between the blue reads and the red reads that you do not observe, in this more domain-like enrichment patterns for sure. Yeah. But there are other tools that you can use, in these situations. This is from a paper, from a base-peak paper, and this is they compared the performance of the approach to the performance of Max, and this goes back to the Motif analysis, right? I mean, they basically said, well, look, if our peak calling approach is good, it should, for a given ChIP-seq data set, of which we know something about the factor, we know what it should bind. The peaks that we find should have a higher enrichment in terms of DNA motifs, so this known motif, compared to another peak caller. So another peak caller should have a weaker enrichment of the known DNA motif within the peak regions, right? And this is kind of what they're showing here. This is actually doing this analysis across many ChIP-seq data sets, and basically showing, this is actually a p-value, so I mean, some of you might have things to say about this, but the Weill scale here is p-value. Basically, what it's saying here is that the enrichment p-values in base peaks for enrichment of DNA motifs within the peaks regions is actually overall better because the p-values tend to be a little bit lower, right? Now, obviously, you can easily tell that the median, which is this thing here, is not anywhere close to 0.05. So it's it's kind of a slightly hand-waving way to compare two approaches. Yeah.
In a case like this, you expect it to be a limitation of the motif enrichment, a limitation of the peak calling, a limitation
Yeah.
of the...
So here in this particular analysis, they did not use de novo motif analysis. So they had a motif that's known to be bound by the factor for which we did the experiment. So they just scanned the peaks and basically counted how many peaks have the known motif that the peak should contain, most of the peak should contain. And for each of these experiments, that gives you a p-value, and basically showing that the p-value is a little bit lower in the peak caller compared to another peak caller. This is not using de novo analysis, so the performance of a motif analysis doesn't go into this figure. Yes?
So you compared all these with base peak with ones that weren't found by base peak but were found by max, but what about the other way around?
Right. Yeah, I know, of course. Yeah. So this is absolutely, this is an analysis that should have been done. Yeah, absolutely. Yeah. Obviously, they were not in the business of promoting max, so they didn't do it analysis. But that's an analysis they should have done if they had been fairly comparing the two methods.
I mean, I guess part of the question is how many peaks you find. If you can be more stringent,
right,
then you can get a better p-value, right? Or hopefully.
Yeah, no. So and that's also the case. I mean, the sample size. If you have more peaks, and this goes back to sample size, and all of that goes into calculating these p-values. If you have more peaks, your p-values are going to tend to be lower, so it may just be that this set has more peaks, and therefore you get better p-values, but it's not, it doesn't translate into an enrichment, like a full enrichment. It's just better statistics essentially. But that's another story. So this is a tool here that we developed called Chipseeker for peak analysis. Just going to tell you quickly a few ideas about it. I mean, it's just meant to again to illustrate the way that ChIP-seq data is analyzed. This is again a nice peak in the genome, very sharp background here, but not too much. But not all peaks are that sharp. So often what you see is that you have is peak-like structures. Likewise here you actually don't see the blue distribution and the red distribution kind of being shifted, right? So, but still, I mean kind of visually it does look like a peak, right? So I mean you would want to detect those. So this is why we actually designed a new ChIP-seq peak calling algorithm. Again, I'm just showing it to you because it's simple and it illustrates some of these statistical tools that you've been using and that you're going to continue using this course. So the way that we do peak calling is to calculate read count at each position in the genome. We really calculate read counts at each position. This is actually exactly what Matt is going to teach you how to do. I think, no, that's what he should have told you how to do later on. But this is basically calculating read counts at each position. Yeah, he's going to show you how to do this in Windows. But and the goal is based on these read counts at each position is to calculate to determine whether the read count is greater than what you expected just by chance. So similar to what Max is doing, but not using Windows, we're looking at each nucleotide one by one. Okay? Again, we need to correct for input DNA reads. Also, one of the issues is typically when you do ChIP-seq, right? You don't want to spend too much money sequencing input DNA, right? So what most people do when they do ChIP-seq, they do multiple lanes or multiple runs for the transcription factors for the actual chip, right? And then they do very few input DNA sequencing. What you end up with is a huge discrepancy between number of reads between chip and input, right? So, and then you have to be able to compare those two very different number of reads. And that brings some challenges. Again, challenges that we've been trying to address here. People, address the challenges in different ways, but this is what we've been doing. So we start with reads. Again, remember the reads are only one extremity of the fragments that are sequenced, right? So they actually don't represent quite exactly the DNA fragments that have been pulled down, right? They represent the extremity of the DNA fragments. So what we do in this approach is to in silico extend the reads. Basically, we extend the reads in such a way that they do match the expected read size that we got from a typical gel like this after sonication. We do that in both directions, right? And then what we do is to calculate a read count profile for each nucleotide. We just count how many reads map at each position, right? That gives you a read count profile like this. Again, each nucleotide we have a number, right? And what you want to do to call peaks, again, in a very simple way, you want basically to look for these regions in the genome where you have essentially contiguously multiple nucleotides that have a read count that's higher than the expected read count, right? So this would represent the peak here, right? This region here. So how would you calculate this expected read count? What's the expected read count in a given experiment? How many reads would you? Yeah, exactly. Yeah. So how would you? Yeah.
Whatever you got in the IgG control.
Say again.
Whatever you got in the IgG control.
You could use that. Let's say that for now you don't want to use that again because you have different read number size. It's kind of let's just calculate what's the expected read count in a given experiment.
Number of reads divided by the genome size.
That's right. Basically, imagine the situation where you were to take all of the reads that come from your experiments and you throw them randomly in the genome, right? If you were to do this and count how many reads map per position per nucleotide in the genome, basically the calculation for this would be the total number of reads. You do have to extend the reads by the actual not only read size but also fragment size, basically, and you divide by the size of the chromosome. That basically gives you the expected read count, right? Would that give you a reliable way to measure expected read count? Why?
Not the entire genome is unmappable.
That's right. That's right. As we discussed, there are lots of regions of the genomes that are unmappable. I mean, most of the human genome is repeats, transposons, and so on and so forth. So that means in practice there are lots of regions where you cannot have any reads, right? Because if you map a read to that location, it maps to 100 different places with the same quality, right? So what that means is that the effective size of a genome is actually much lower than the actual size that you get in the reference. Okay, this is the sort of effective mappable size that we care about. We'll get back to this, but. So basically, once we have a read count, and once we have an expectation, right, we can just like showed you before, just feed these two numbers into a Poisson distribution. I mean, before you want to show that the read counts do follow a Poisson distribution, right? That's the case for this experiment here. But then you just simply put this read counts into a Poisson distribution, and that tells you, that actually answers the question is whether the observed read count at a given genomic position is greater than expected by chance, right? So you put this into this function here. This is the kind of numbers that you get if you were to feed all these numbers in there. We do take the log 10 just because of, you know, these numbers are very small, so, you know, it's just easier to manipulate in the program, in the computer program, if you take the log 10, log base 10 of this. We do also take negative log just as a way to invert the sign. And again, this is something that you can do, you know, very simply in R if you wanted to, just using the Poisson distribution. And what we do effectively is to convert this read count profile here across the genome into basically a negative log 10 p-value profile of the genome. Right. Now the nice thing about this is that we can do exactly the same procedure in the input with the input data. Right. Remember, they are on very different scale, right? But if you do the same procedure in the input data, you know, the expected read count is also going to decrease if you have less reads, right? Or at least, you know, this is, yeah, the expected read count is going to decrease, right? So what that means is that once you convert these read counts into p-value profiles, then the p-value profiles of the ChIP experiments and for the input experiments are going to be on comparable scales, right? And therefore what we do is to simply subtract one from the other. So essentially removing one p-value, one log 10 p-value from another, right? And that allows you essentially to reduce the size of the peaks in the ChIP data using information about the input data, right? And now you are in a sort of controlled, you're in a sort of normalized situation where you can look for contiguous nucleotides whose difference between log 10 p-values is above a threshold, right? And we look for these regions here, right? Basically, it's equivalent to using this ratio as a threshold. It's essentially a ratio between p-values that we use here. So we calculate this and we use a threshold in terms of detection. Now, the very nice thing about this approach is that it works if you didn't do input DNA sequencing. If you didn't do input DNA sequencing, and basically your read count in input DNA is 0, right? If you were to feed the 0 here into this function here, right? You go from x, you have x equals 0 here. So you feed this into this function, you have a sum here. So you go from 0 to x minus 1, which is basically you never enter this function here, and that gives you a p-value of 1, right? And so practically now, if you don't do any input DNA sequencing, what you end up with is a situation where this here, the denominator is always 1, and therefore you can still use the same framework essentially to detect peaks. It's just, and now you're not normalizing that by the input DNA, right? It's nice because the same tool, the same approach allows you to do peak detection whether you do input DNA sequencing or not. And again, sometimes, especially for transcription factors where, usually get very, very sharp peaks, sometimes people don't really do input DNA sequencing. A peak, is going to look like a peak. They're going to be unmistakable, very easy to detect, and people just don't do input DNA sequencing. They should, but they don't always do. The nice thing about this approach allows you to cope with both situations easily. Okay, so this is mappability. This is again illustrating some of what we discussed about regions of the genomes that are non-mappable. This is one region here. The UCSC genome browser tracks, as we discussed last time, has very nice tracks that you can use to get a sense of mappability across the genome. as you can see here, this is a region that has very low mappability, which is still inside a gene, but it has very low mappability. Most likely because there is a repeat here that's identical to another location in the genome. So just practically speaking, what we do is to estimate mappability, either using a genome browser track or using our own k-mer counting across the genome. We can calculate the unique or non-unique fraction of the genome, and that allows us to correct the chromosome length by this mappable fraction of the genome as a way to correct the expectation here. This is just to give you some results. Also to give you, to show you that we do some filtering. We do merge peaks that are very close to each other. If you have two peaks that are reasonably close to each other, we think that they probably should be merged into a bigger one. If a peak is too short, usually it's usually not a very good peak, and we remove those as well. But the simplicity of this approach means that you can process millions and millions of reads in just a few minutes. Not quite like the approach I will show you in the lab tomorrow, which is more sophisticated, but also much more slow compared to this approach. This is just an application here to a transcription factor, and this is just to give you a sense of what these peaks look like. You can make you get essentially a bed file out of these tools, and you can upload the bed file onto the genome browser. And that allows you to visualize your peaks in the context of the reads, for example. And when you do these experiments, again, this is kind of a number of peaks that you typically get, from ChIP-seq, at least for transcription factors. And usually, again, the peaks, tend to be close to genes, not necessarily too surprising. These are some of the tools that we have. We have a whole framework that allows you to do many, many different things using from ChIP-seq data. And it's not yet an R package, but as we discussed multiple times, it's reasonably easy now to integrate command line within your R package. This is an example here where you would run Chipseeker, that's a tool we have, using the system command. It would produce a file of peaks, and you can load this file of peaks into R and create very easily a range data or G-range object using a peak file that will come out of this tool. Or again, a lot of tools will produce this kind of bed file-like format that you can load into R very easily. Okay, just to make a point that for certain operations, R might not be ideal. You might want to use an external tool like this. It's just faster, essentially. But you can load the result into R and basically have one workflow that just does everything for you, without having to leave R at any time. Okay. This is a non-exhaustive list of other peak finders. There are actually lots of them now. This paper actually looked at all of them and compared the features. Some of them are better than others. Some of them better for certain things and worse for other things. But this is just to show you that there are definitely situations where you don't want to call peaks, especially we discuss situations where the enrichment is weak. You don't have a very strong peak shape across your genome. That definitely applies for histomodifications like H3K27 transmethylation. An alternative to calling peaks is to quantify within a given windows, for example, windows around the transcription start site, to quantify peak height, quantify essentially redensity in those regions. And you can use that to compare conditions. You can use that to compare experiments and so on and so forth. Not calling peaks, just quantifying how many reads you have in a given region. This is a very powerful way to analyze also some of these non-peaky ChIP-seq data sets. Okay. So peak analysis interpretation, these are the set of operations that you want to do once you get peaks. I usually say, getting peaks is obviously, to me, it's actually the most boring part of the process. Peaks don't really tell you much. It's the analysis of these peaks that's really helping you interpret your data. The multiple things that you can do with your peaks. One is looking at where they fall in the genome with respect to genes. So this is just for one experiment here, giving you an idea of the distribution of peaks across multiple regions that are related to genes. So you have promoters here, which are defined as 4 kb window centered on transcription start site. You have downstream exons, introns, distal, intergenic, and so on. And this is the distribution of peaks. For this particular factor, distribution can vary between factors, but it's useful to have to show this. And this is actually one of the exercises that we'll have you do tomorrow. Build a pie chart of peak locations for a ChIP-seq experiment. Integration of multiple peak lists is something that a lot of people want to do. Typically what you do is to do multiple ChIP-seq experiments. You get multiple lists of peaks, and you want to essentially compare the location of these peaks to look at which peaks are overlapping, which peaks are unique to a given data set, and so on and so forth. And there are some very useful tools in R to do this, like G-range or range data, some of which we showed you last time, and we'll continue to show you tomorrow. Conservation analysis, this is giving you a sense of how conserved the peak locations are on average. This is a profile of conservation. This is looking at enhancers. This is some analysis we did at some point. This is looking at conservations of enhancers, or windows centered around enhancers, focusing on 163 enhancers. And you can see here there's a very sharp peak of conservation. These are enhancers, they're outside of genes, they're far away from genes. But despite that, you have a very sharp peak of conservation. This is also, it can be used as a quality control measure, right? I mean, you do expect that your peaks, even if they're far away from genes, assuming that many of them are functional, that the conservation across species would be significant and very high, and that's usually what we see. What's important though is to always do a comparison with background, right? So here, the background is basically 163 regions that are chosen randomly across the genome, for which we also estimate conservation, the average conservation. As you can see, the conservation is uniform across the window here. You can do that using, you can do this analysis using some of the genome browser tracks, which you can also load into R. This is one of those genome browser tracks that has conservation values. This is just values between 0 and 1, and 1 is more highly conserved and 0 is less conserved. Yeah. So fastcones you don't have to run the tool. If you go to this address, and there's one for G19 and for MM9, you can download these tracks here that have single nucleotide conservation measures. So the way they measured conservation across species, that's done using a hidden Markov model, using a phylogenetic analysis in a hidden Markov model, essentially comparing two scenarios, high conservations versus low conservations. The probability of being in the more highly conserved scenario. But this is what this is showing here. And this is per nucleotide, so each of these value here corresponds to a nucleotide. I mean, the way that the fast constructs are working is that it works by intervals. I mean, and the reason is that some regions of the genomes are hard to interpret in terms of conservation. They might not have an equivalent or homologous regions in the other genome, so you don't have data for the entire genome, and therefore you have windows like this. But this is telling you that this is a window here, chromosome 1, that starts at this position. The step is 1, meaning 1 nucleotide, and this is this position here, this is the next position, and so on and so forth. And that basically contains information about every nucleotide, the conservation of every nucleotide in this window here. And you can get conservation profiles, across multiple, clades, primates, mammals, all species, and so on and so forth. Okay. So another thing that obviously is very important. Yeah. Sorry. Yeah. Yeah, sure. Sorry, thank you for reminding me every time. Yeah, no, okay, so yeah. I would just continue and never stop. So I have a few more slides, not too many, getting close to the end. These are a couple of slides about motif analysis from ChIP-seq data. Obviously, you're doing ChIP-seq data for a given factor, and one question that you often would want to ask from your ChIP-seq data is, what's the code, what's the DNA code, what are the DNA sequences that allow the factor that you did ChIP-seq for to recognize, to help essentially the factor bind to the DNA in a specific manner. The other question that you would want to ask is, are there any, can you use ChIP-seq as a way to detect combinatorial regulation, right? I mean, you're pulling down a factor, right, when you do ChIP-seq, but that factor is often going to bind together with other cofactors or factors that might also bind to the DNA in a nearby location, right? And you would think that when you look at the sequence of the peaks, you might be able to detect also the presence of these cofactors just through a DNA motif analysis. DNA motif analysis, in addition to also the QC step that I mentioned, is very important when it comes to ChIP-seq. There are multiple approaches for this. One is the meme approach. This is a reasonably old, but still very frequently used way to look for motifs. It's using a sort of an EM, expectation maximization approach for motif discovery. It's a probabilistic model essentially that allows you to look for motifs using a definition of a background sequences, essentially looking for motifs that don't look like a background sequence definition that you define. This is a very again a very well established tool for motif analysis that has been extended in the last few years with a lot of tools that allow to analyze what comes out of a motif discovery procedure itself. Some of the most important aspects of this comparison with known motifs. So when you do a ChIP-seq analysis, a motif analysis, you're going to find motifs, and really one key question that you want to ask is whether these motifs look like anything else that's been found by other people in terms of DNA recognition site, right? Again, when it comes to identify cofactors, that's very helpful. When it comes to really understanding, the code of the factors that you pull down, that's very useful. And a tool like TomTom, which is integrated to meme, allows you to do these comparisons, the scanning of databases. Also, some of the tools that are in this framework here, which is available at this address, allow you to do pathway analysis of the peaks that have a given motif as a way to understand and to dissect, further the biology of the peaks. I do want to tell you about one thing that we did a couple of years ago in terms of motif analysis. There's a paper we published in 2007 that is actually quite highly applicable to ChIP-seq motif analysis. This is a procedure for unbiased motif analysis, motif discovery. Again, and by unbiased, the de novo idea is that you're not using a database of motifs to do a motif analysis. You want to discover what's in your sequence, what's highly abundant in terms of short sequence, short motif within your peaks, right? And again, the key aspect about this type of analysis is to define the problem in a very careful manner, right? And when you say, I want to find short sequences that are highly enriched in my peaks, right? You have to keep in mind that you have to define what that means by what you mean by enriched, right? Usually enrichments means that you're going to compare your peaks to another set of locations in the genome, right? And you want peaks, you want motifs that are more abundant in your peaks compared to these other locations, right? So how would you extract these background locations in the genomes? What would be a good set of background sequences that sort of match your peaks in terms of certain characteristics? How would you define the background for these for motif analysis like this? Again, you want to find peaks that are in your sequences, but not in the background on background sequences. How would you define these background sequences? How would you do it? Yeah.
Could you just take the
Okay. So what you're saying is basically the background would be the rest of the genome.
Well, including.
Right.
With your placement.
Including
Including.
including the peaks, your peaks, right? Yeah. So, okay, I mean that's that's a possibility. The only thing is that if you were to do an analysis, discriminative analysis where you look for peaks that are in your sequence and less abundant in the rest of the genome, I mean you would have to scan the entire genome many times, and that would basically take forever. Right. So is there a better way to do this? So you have a set of peaks, and what you're saying is that what you could do is to randomly select peaks of the same size from the genome.
That's one option.
Would right? Okay, so exactly. So you have to be careful about this, right? Because imagine a situation where you have your peaks that overlap with CPG islands, right? Let's say that you have half of your peaks that overlap with CPG islands. Well, that means that your peaks are going to be very GC rich, right? So now if you select the random peaks, sort of background peaks, completely randomly in the genome, because CPG islands is such a small fraction of the genome, most of your random peaks are not going to overlap with CPG islands. So when you're going to do motif analysis, the only thing that you're going to find is a very strong CPG signal. That's not going to tell you precisely, what kind of code. I mean, it's going to tell you something about the overlap between your peaks and CPG islands, but it's not going to tell you more than that, because the signal is going to be overwhelmed by the CPG islands. So is there, is there another way by which you would select these random peaks? I mean, you kind of said it. You essentially want to match the CPG content of the dinucleotide content of your peaks.
But in principle, you could also just take if you have enough, just take your sequences, you randomize maintaining.
You can do that, but then again, it's this is good because this is things that people do, right? The problem is that if you randomize your sequences, the random sequences are not in the genome; they're fake sequences. So now the question that you're asking for, are there any motifs that are in my peaks, that are more abundant in my peaks, but less abundant in those fake sequences, right? It may be a valid question to ask, but it is a question that is potentially different from another question that you could ask. It's comparing with a fake version of a human genome, right? So another way to go about this would be, for example, to take a peak, right, and use as a background the adjacent peak location, right? You have a peak, let's say, of 200 nucleotides. For each of the peaks, you're going to take the background, the adjacent 200 nucleotides window, right? And you're going to do that for each peak. So for each peak, you're going to have a background that's defined by the surrounding DNA, right? And that's actually going.
And the CPG island is short.
Yeah, but usually, yeah, exactly. I mean, you can have this, definitely there's this potential, but in most cases, it actually should correct for the fact that you're in CPG-rich regions, and the background peaks would, have the same CPG island overlap as your peaks, right?
Well, I wonder if you could use your control samples, take peaks out of the pseudo peaks that are control samples.
Yeah, I mean, in theory, you shouldn't have too many peaks. That's the thing. And these peaks will be also highly biased because we tend to be at promoter regions. And so the composition of promoter regions is different from the composition of the rest of the genome. So again, if you were to use this promoter pseudo peaks as a control region, you would ask a very specific question. Is there something that's different between my peaks and promoter regions? It may be a question that you want to ask, but it is a very specific questions with a very specific biology attached to it, right? The point of this, I want to make you understand that the definition of a background is very important, and it really should be part of a specific question that you're asking about your peaks, at least in terms of motif analysis, okay? So let's assume that you've solved this problem, right? That you have you specified the question very clearly, right? And that indeed you're able to identify a set of random regions that share a lot of characteristics and features with your true binding peaks, right? So this would be your true target regions, this would be your random regions, right? And you have a label that tells you whether a region is a true region in the genome from your ChIP-seq analysis, or if it's a random region that you selected randomly, okay? Now, the idea is that you would want to look for motifs like this. This would be just representations of motifs, right? That are more highly abundant in your target regions compared to the background regions, right? So this is a situation like this, right? There's a higher abundance, a higher fraction of peaks that have a motif in this group compared to this group, right? So as it turns out, it's part of a significantly larger framework, but there is a way to phrase that problem here. There's a way to phrase it as an information theoretic problem. And the way to phrase this as an information theory problem is to ask whether the presence of a motif within a peak tells you something about the label of a peak, whether it's a true peak or a random peak, right? Is there information about the location or the presence of a motif in a peak and the label of a peak, right? There is a way to address that question, to quantify that correlation, and that's using the mutual information. I think that's something that you've heard about today. It's basically a very broad way, very general way to define correlations between two variables. The two variables here in this particular situation being presence or absence of a motif, and what is the nature of a peak? Is it a true peak or a random peak? Okay. The point here is that you have two variables, and you can calculate the mutual information between two variables, the correlation between these two variables, and you can the way to calculate the mutual information is using this function here. The basic idea of this function is really contained in this term here, where if you have two variables that are independent, right? The probability of the ratio, the product of the probability of each variable is going to be, so the joint probability of the two variables is going to be equal to the product of the probability of two variables. So this here is going to be equal to 1 if the two variables are independent, right? And the log of 1 is going to be 0, and therefore the correlation of mutual information is going to be 0. So two variables that are independent that don't say anything about each other, you get an information value of 0. Yeah. If the variables are independent, if one variable tells you something about another variable, this function here is going to give you a high value, and therefore you're going to have a high correlation. And this is what we use as a way to detect motifs. Now again, it's part of a broader framework where you can really generalize this notion to not only two classes of regions, right, but many, many classes, right? You could imagine that you do many ChIP-seq experiments. Each peak now corresponds to one different factor, right? And you could want to get a sense whether there are motifs that tend to be enriched in one set of peaks and not in another set of peaks, right? It's the same idea. It's really kind of containing the same idea about information between motif presence and absence across peaks and the labels of those peaks. Again, you can really, really generalize this notion to multiple categories and not two categories. The application of this analysis to two categories is basically discrimination between two sets of sequences. And this again, applying this formula here, it just involves calculating the joint distribution between the two variables, between the motif presence and absence. Can anybody say how is this calculated? How do you calculate this number here? Where is this number coming from? This is a joint distribution of motif presence and absence and the peak label. How do you calculate this? What goes into calculating this value here? It's based on this figure here. All the information is in that figure. So it's joint distribution, right? So you have a first variable which is motif presence and absence. If you have a little thing like this, a red thing like this, it means that the motif is present. If you don't have it, it means that the motif is absent. Okay. So how do you calculate this? How do you calculate this joint distribution here? You just count essentially. So what goes into counting here?
How many of the yeses have a motif?
Right. So how many? Basically, with this entry here is about, it's about the number of cases across the entire set of peaks here, where you have both situation where the motif is absent and situation where the factor of the peak is not a true peak. This is one of these nodes here. Right? So you just count. This is, in this particular peak here, the motif is absent. It's one. It's also belongs to the non-peak category. Okay, it's one. Another one, 2, 3, 4, 5. Okay. So you have 5 that are in the motif absent and the non-true peak category. What do you do with this 5? You don't have a 5 here, so what do you do with 5? That's right, you divide by the total number of sequences. That's right. This is how you build a joint distribution. Right? It's very simple, just counting essentially, and dividing by the total number of elements that you have in your situation. Right? So you build this joint distribution here. Just apply this formula here. This is again this is an entry in the matrix, right? This is the sum of a column or a row of the entries in the row and our columns. And you just apply this and you get a value. And if the value is high, it means that you have a strong correlation between your motif and the labels. If the value is low, it means you have no correlation. They're kind of random with respect to each other. Okay. So that is a way, again, more general way, but I don't have time to go into details, but it's a very general way to look for motifs. Now, this is what I showed you so far is just a way to determine whether a motif is good or not. If a motif is good, it should have a high information value. If it's not good, it should have a low information value. Okay? You know what a good motif looks like at this stage. So now the question is, how do you look for good motifs? Again, a motif being a short sequence, 7 nucleotides, let's say. How do you look for it? You know what a good motif is like. A good motif is this. Okay? Now, let's say that your motif is 7 nucleotides long. How many sequences of 7 nucleotides can you have? Yeah, that's about 16,000, right? So what do you do? How can you search the space of motifs?
Same way we just were talking about earlier?
Right, absolutely, yeah, absolutely. That's right. So basically what you can do is to enumerate all of them. You have 16,000, it's actually not a long, it's not a very long list, right? It's something that is absolutely tractable computationally. So you can look at all of them. And that's what we do in this approach. For each possible k-mer, so this is, we use k equals 7 as a size of a motif. We calculate the mutual information value. Right? So we have 16,000 information values. Right? Again, each of them is calculated using this approach. It's really exactly this approach here. Exactly this the counting procedure that we did together. That's what this algorithm is doing. Nothing else. Okay? You get information value for each of the k-mers, right? And you look at the k-mers that are at the top, right? And according to this analysis, these should be the k-mers that are most highly enriched in your peaks, right? Now, is there another way that you could get a strong information that would not involve enrichment of motifs within your peaks? Is there another way in which there will be a non-random distribution of motif with respect to the binding peak labels? Right. What about motif depletion? Right. What about a situation where you would have none of these true peaks that have a given motif, and let's say 50% of the random peaks that have a motif? Right. That would be a non-random association between the motif and the peaks. Right. So that would be something that this procedure here would pick up. Right. And that could be meaningful. Right. So what could it mean? you have a motif that's even less abundant in your peaks compared to the rest of the genome. How could you get such a situation?
Pressing factor for your proteins.
Yeah, that's right. It's basically a motif that you do not want to have around in the context of binding of a given factor. So you don't even you want to have it around even less than random, essentially. So basically, it would be a binding site for a protein whose function would be detrimental to the function of the factor that you assay for binding. We've seen this many times. It does happen all the time, right? That you want essentially to exclude certain motifs from a particular location, so that whatever binds to this location can do its job without interference from other things. Okay. It happens all the time. The point here is that this technique would pick up these two signals equally well. And overall, so I'm not going to go into too much details about this, but obviously k-mers is not necessarily a great way to represent motifs. The other issue with k-mers is that, and you see it here, right? I mean, sometimes, you have two k-mers. These k-mers are pretty much equally good, right? They're just shifted, shifted version of the same, right? So you would want to merge them into one. It basically means that the motif, itself is probably a bit longer than 7, because two shifted versions of the same motifs are almost equally good. But the point here is that in this procedure that we have, we actually don't stop at the k-mer stage, just because, again, you've seen that with Harman's lecture today. There are better ways to represent motifs. I mean, proteins that bind to the DNA, they don't bind usually to a single k-mer, right? They bind to, they have a favorite sequence, for example, this guy here, but they also typically bind slightly different version of that sequence, right? So k-mers in that situation is not a good way to represent that degeneracy, that flexibility in terms of binding. There are better ways to do this, and weight matrices is a good way to represent this flexibility in terms of binding, at least at certain locations. Usually in the DNA motifs, you have certain locations that are very strict. You need to have a particular nucleotide, but there are also positions where you can have, you can choose between a small subset of nucleotides, and with a little effect on the affinity between the binding and between the DNA and the protein. Right? So the point being here is that we can take these k-mers here and change these k-mers, essentially mutate these k-mers in such a way that we can get essentially more reliable and more accurate, and most importantly, more informative versions of the same motifs. It's just essentially better representations of the motifs, and we do this by trying to improve the mutual information even more by making point mutations into these k-mers and trying to get to weight matrices.
Yeah. I'm not trying to understand that, because those other point mutations are already on the list. So
yeah, exactly. So another strategy would be to say, okay, look, I have this sequence here, right? I'm going to look down the list, right, for things that are similar. I'm going to try to combine these two k-mers into one, right? And that would still, potentially lead you to something similar. It would just be another way to get to a more informative motif. This is the algorithm that we have is using these point mutations, but you're right that some of this information is down the list. We actually don't do it this way, but it could be used as a way to constrain the exploration a little bit more.
So when you say you make point mutations, you take that top one and then you just isolate it and just change it one at a time and see. That's exactly what we do.
Yeah, that's exactly what we do. And again, it's absolutely linked to the fact that we have a very good way to define motifs, right? So we have an objective way to define whether a motif is good or not. So if we make a change in the motif definition in such a way, for example, that you would have a new motif match that occurs here, and this peak that did not have a motif before, the information would actually get increased, right? And therefore, according to this criterion, it tells you that it's a better motif, right? And we would keep that motif and continue to mutate that motif in such a way that it keeps getting better and better. When we can't make any changes to the motif as to make it better, when basically, there's no new definition of a motif that would, better separate, these two sets of regions, we kind of stop and say, okay, this is the optimal motif, and that's it. Yeah, so this is an illustration of what you get at the end. This is actually an example. So all of these yellow motifs here are motifs that are highly enriched in your set of peaks of interest. But just like I told you, very often we do see motifs that are depleted from your peaks. And again, that sometimes carries some interesting biology, and you can learn something by looking at what's depleted in your peaks. We also do co-occurrence analysis that goes back to this idea of looking for, trying to understand whether there are cofactors for a given motif, for a given factor, based on the motif analysis. This is looking at the extent to which, so these are all motifs that we find under a given set of peaks. You see that there is definitely some variability. This is actually a BCL6 ChIP-seq analysis here. We did find the BCL6 motif, right? But in fact, the strongest motif in this analysis was not a BCL6 ChIP-seq, BCL6 motif. It was actually an ALK1 motif. It's an ETS family type of protein that is known to bind to this type of sequence here. But the point here is that all of these sequences came from the same set of peaks, right? I mean, they're not present in 100% of the peaks. They are essentially more abundant in the true peaks compared to the random peaks, right? And this sequence here is more abundant in the random peaks compared to the true peaks, okay? The point here I wanted to make is that you do see several motifs, right? And when you look them up, I mean, you see that they do belong to, they've been seen before, and they do belong to different families, essentially the different, bound by different proteins. But now what you can do is to look at the extent to which these pairs of motifs essentially occur within the same peaks. Because it could be that you have multiple motifs that are enriched, right? But it could be that 50% of your peaks have a certain motif, and the other 50% has another motif, right? So we do want to get a sense whether these motifs tend to co-occur within the same peaks, and these are analysis that can be done as well using similar concepts of information. But the point here is that whenever you have an entry that's, this is a pairwise matrix here, but whenever you have an entry that's white, it tells you that two motifs likes to be in the same peak. They tend to be found more highly than expected by chance, more frequently than expected by chance within the same peaks. When you have a plus here, what it tells you is that not only do the motifs like to be in the same peaks together, but they're also closer to each other in these peak regions than what you expect by chance. But also tells you something about the tendency of the proteins to bind to these motifs to actually work together potentially as a protein complex, factor and cofactors. Okay. Again, the point here is that by doing this motif analysis, you can actually reveal a lot of really interesting biology about your ChIP-seq experiment. Not only by the motif enrichment, but also by the co-localization of motifs within your peaks. Yes?
I kind of have a technical question, since if you are looking for your ChIP is against a certain protein, then you look for cofactors.
Yeah.
Do you think it's necessary to extend your peak region a little bit?
Yeah, so that's definitely a good question. The peak regions, remember, are already pretty long. So the protein-DNA interface, right, is very short. It's just a few nucleotides long, right? The peaks, the sequence that you pull down, and therefore the peak regions that you detect, tend to be much longer than the actual protein-DNA interface, right? So if you just take the peak width that just come out of the algorithm, you already include a lot of flanking regions, and therefore you include all of the potential for additional binding sites on these locations. But there are other things that you can do. For example, if you do ChIP-seq for a transcription factor, and if you do in the same cells ChIP-seq for histomodifications, like for example enhancer associated modifications, like K4 dimethylation, K27 acetylation. You can use that information as a way to identify the enhancer region. You would have essentially the binding factor that binds, and you would have potentially the enhancer regions a bit longer, right? And that would contain potentially additional sites for your cofactors. Often when you do this, you see that the binding factors actually binds between two nucleosomes. And, and it's not clear whether there's any cofactor that binds on these two nucleosomes. So you could use that information as a way to remove the nucleosomes and focus on the naked DNA. That's where the motif action potentially would be. So there are all these tricks that you can use as a way to restrict the search for motifs within your peaks. Anyway, so just quickly, briefly, do you want to tell you about a couple of papers that interesting? So this was the first ever ChIP-seq paper. A few years old now, but did ChIP-seq using for NRSF and other elements. Just in kind of a landmark paper in the sense that it's the first ChIP-seq paper. This is the first ChIP-seq paper that actually defined these enhancer regions I was telling you about, and defined the fact that enhancer regions are actually marked by specific histone modifications. Enhancer regions are these regions that are far away from genes, that have to loop over probably the genes, using chromatin looping as a way to affect gene expression. In this paper, people realized that when you look at these enhancer regions, there's, even though we are actually very far away from genes, there's a signature for these enhancer regions. And they found that basically there are two histone modifications that are important. K4 monomethylation tends to mark those enhancers, and K27 acetylation is also a mark of enhancers. When you have these two marks together, you essentially have enhancer regions. And that's actually a very powerful way to detect these enhancers. Again, keep in mind that they can be very far away from genes, right? And it's very hard to do these looping experiments, where you can tell whether a region of DNA that's very far away from a gene is actually looping into a promoter. This is doing ChIP-seq for these two histone modifications is actually a very powerful way to detect these distal enhancers. Since then, people have realized that there are multiple classes of enhancers. There are active enhancers, poison enhancers. Active enhancers are essentially the ones that have these two histone modifications I was telling you about, K4 monomethylation, dimethylation, and K27 acetylation. There is a class of enhancers that are poised, essentially, that are ready to be activated but not yet active. These are the enhancers that have only monomethylation or dimethylation. These two histone modifications are very similar to each other. But these poised enhancers do not have K27 acetylation. So there's a multiple class of enhancer regions. I mean, this paper took this a little bit further and look at combinatorial marking across the genome. They did ChIP-seq for multiple histone modifications, and basically used a segmentation procedure to segment the genomes into regions that have the same composition in terms of these histone marks. And they found regions that they define in multiple ways. For example, active promoters that would have K4 dimethylation, K4 trimethylation, K27 acetylation, in multiple classes of regions that can be classified by looking at the histone modifications. And that's using an HMM model to define those regions. This is just to show you that you can do other things. This is not really using ChIP-seq, but it's trying to get a sense of what's happening on the chromatin. You can do nucleosome positioning, mapping in the human genome. The way you do this is, so nucleosomes are these multi-histone complexes, right? The DNA is wrapped around the nucleosome. The amount of DNA that's wrapped around the nucleosome is very fixed. It's about 146, 47 base pair, okay? One experiment that you can do is to digest the DNA that is naked, that is not wrapped around the nucleosome. There are enzymes that allow to digest a non-wrapped DNA, right? So that's what they did in this paper. They digested this some naked DNA. They're left with DNA that's wrapped around nucleosomes. They get rid of the nucleosomes and they sequence the extremities of the fragments that were wrapped around the nucleosomes, and they can map that back to the genome, and that basically gives you the locations of individual nucleosomes on the genome. And this is these are actually experiments that we did in my lab in collaboration with another lab at Cornell. But these are actually single nucleosomes here. This is using pattern sequencing of the two extremities of the longer DNA fragments. And you have an accumulation of these reads here that map to the same location, and that basically marks individual nucleosomes. So this would be one nucleosome here, another nucleosome here. And I always found this really cool because this is almost like a microscope, right? You can basically see the nucleosomes, on the genome. You can see the valley here between two adjacent nucleosomes, right? And when you do this, this is what I was telling you about, the fact that when you look at the binding of a transcription factor, and when you look in terms of nucleosomes, you very often see that factors bind between two nucleosomes. This is essentially the naked DNA portion between two nucleosomes that's accessible for binding by transcription factors. These are two well-positioned nucleosomes on either side. This is an average picture across many, many peaks in the genome. I guess that's it. I'm happy to answer any more questions that you'd have. Yes.
This is a biological question.
Yeah.
Might be very
Right. Yeah.
naive or very silly, but so essentially transcription goes through the DNA that is still bound to the nucleosome.
Yeah. So that's a good point. So the whole genome is wrapped around nucleosomes, but in the RNA polymerase is transcribed in genes, right? I mean, you'd think that would be bothered, by these nucleosomes, or they're all over the place. I mean, what's happening is that the RNA polymerase is not coming to alone. It's coming with essentially a truck of proteins. Some of the functions of these proteins is to kick out nucleosomes that are on its way. So the RNA polymerase is able to just remove nucleosomes as it moves through the genes, so that it can do the transcription in a smooth way across genes. And the nucleosomes reform right after the RNA polymerase is passed. Yeah.
But so in principle, you could follow transcription by this repositioning.
Yeah, I mean, it's remember actually the speaker, the Nick, right? I mean, showed you how we could use sequencing as a way to get some information about some kinetic parameters of the speed with which ribosomes go through mRNAs, and you can do somewhat similar things using some of these techniques. We can also actually do this using RNA-seq. People looked at the amount of mRNA that maps to a given gene. It is actually kind of a slope. If you look carefully, because you have multiple RNA polymerase across the entire gene, there's actually there's a gradient of mRNAs that are produced for a given gene, and you can use that as a way to deduce kinetic parameters of how fast the RNA polymerase is going through a gene. This is not using these techniques, but it's
How can you use PCA for this purpose?
yeah. So this is just a decomposition. So what we did here was to look at each window. This is actually centered on the transcription start site. The transcription start site would actually be here. We looked at 10 nucleotides windows. Okay, in a window, 10 nucleotides is intervals. In a larger window, this is -1 kb. This is the transcription start site, and this is +1 kb here. Right? So we have these 10 nucleotides windows. We count how many reads we see in each of the nucleotides on average. Right? That gives you a signal, and we do that for every possible promoter. Right? So you have a profile line for every single promoter. What we did here is a decomposition, essentially, is a principal component analysis of these profiles of nucleosome binding. Right? And when we do this, what we see is that there is a very strong component, which is this. So this is kind of nucleosomes all over the place, not so much interesting. This is the one that's interesting. So basically, this is a principal component. This is a pattern, essentially, that's very strong in the data. That's what principal component is doing. It's a pattern that's very strong in the nucleosome profile that corresponds to high nucleosome occupancy right downstream of a transcription start site. Right? This is basically, and in fact, there are lots of genes that are like this. So in fact, genes that are active actually are shifting. They have this +1 nucleosome. It's basically a nucleosome that's well positioned right downstream of a transcription start site. Right? And in fact, when you look at genes that have this kind of pattern in terms of nucleosome localization, these are highly active genes. Right? This is another pattern here where this is a class of genes that have a nucleosome that's not shifted. It's basically right on the transcription start site. These are genes that are not transcribed. These are genes that are inactive. The nucleosome is there, and this is a configuration that's not prone to activation. So it allows you to identify all these different patterns here of nucleosome binding, and again, some of which corresponds to different classes that you do see in a given across a genome. Yeah, that's it. Yeah. This is essentially using PCA as the traditional way, sort of deconvoluting multiple patterns within the data that together do a good job at capturing the variability in your patterns. Questions? No? Okay. Yes.
So the fire method you described and the geometric means method we talked about this morning seems like before the n gets pretty big pretty fast. Is there kind of an upper limit to?
Yeah. So this is not the application of a fire approach to motif analysis, the way it was done here, is actually not using the hypergeometric distribution. But
yeah, the two methods.
Yes, that's right. The two methods have definite similarities, and in fact, at some point, we actually did it. We actually looked at the extent to which the hypergeometric distribution and the mutual information will actually give you similar quantities or correlated quantities. And if you do that, you see the line is actually perfect. There's a very strong relationship between the two approaches. Yeah, I'm sure there is a way to explain this question correlation. The point being here is that you are not in the situation. Let's go back to the formula. Applying this formula here does not involve any factorial or any problems, with numerical accuracy. You do have problems in terms of numerical accuracy with a hypergeometric distribution because you have these huge factors that just essentially blow up really quickly. Not to say that this is really a problem, because it's actually reasonably easy to. One thing that you can do is to work in log space, right? And therefore, pretty much all of your numerical accuracy problems when it comes to factorial, go away in log space, because products, become sums, right? So you don't have problems anymore. And there are also ways to calculate the hypergeometric distribution reasonably, quickly, without having to fall into these numerical problems. It's just, technicality, but yeah. And certainly, you do not have that problem here for sure with this formulation. Yeah.
But can you extend it to like 146 nucleotides? Or K equals 146.
Oh, so this is another problem. This is the problem of the size of the motifs, and in fact, the problem is it's distinct, but it is a real problem. If K increases, the space of possible motifs does increase, reasonably quickly. That being said, the space of possible motifs that are also in your genome does not increase as quickly. Because obviously, you don't care about K-mers that are not in your genome, right? So you could build a list of K-mers that are in your genome and only explore that space. And in fact, you would probably want to explore K-mers that are present at least a certain number of times, 10 times. That does restrict your list of K-mers very quickly.
I'm not sure if I understand this correctly, but you start from generating all those K-mers, then look for them in the genome, and then narrow down the list to those that occur more or less frequently.
Yeah, that's right.
Finally, we then map those, and select only those that are similar to the motifs found in Jasper.
That's right, exactly. That's absolutely. We have there's a simple way to. So these are the motifs we get. And you see they have a name here, and what we did was to map these motifs to motifs in Jasper or TransFac. Yeah, that's right.
In the end, if you choose only those that map to those motifs.
Well, we don't choose only those. And as you can see here, this guy here does not have a match in Jasper or TransFac.
Because the question was, why didn't you just start from those motifs?
Oh, absolutely. Yeah, this could be very valid. Absolutely. And a lot of people do that. This is not what we do here. We do an unrestricted analysis. It so happens that many of our motifs look very similar to things that have been found before. And this is, usually good news. But, it's also good news when you have things that don't map to anything that's known before, because it tells you that potentially there's some new biology there and that, could be investigated. Although, this is a difficult problem. Yeah. It's more than that, there's some information on the side. But, going from this, not knowing what, this is bound to to actually identifying, you know, what the binding factor, you know, and understanding the biology of his motif. This takes experiments, you know, it's complicated, but it's doable, yeah.
What proportion of transcription factors would you say we know the binding motif?
So yeah, I would say a significant fraction. So let me qualify this. So factors, transcription factors come in families, right? And by and large, you know, now at least one member of each family is being characterized in terms of specificity, right? And the idea is that very often, you know, it tells you something about the other members, right? Usually the members of the same family of transcription factors bind to very similar sequences, right? So if you have a factor, you know, for which there is another factor whose specificity is known, I mean, you can reasonably transfer specificity from one factor to another. I would say, you know, for most of these families, you know, this specificities are reasonably well known. Now, obviously, there are families of transcription factors that are a bit more complicated, like zinc fingers. So zinc fingers are, you know, messy. Zinc fingers are messy because, you know, you have zinc fingers. You have a lot of them in the human genome, right? And what's happening with zinc fingers is that each finger in the zinc finger protein can recognize nucleotides by itself, right? Basically, free nucleotides, right? And so what the zinc fingers are doing is essentially, you know, they have duplicated the number of fingers within them. So you actually have zinc finger proteins with 15 fingers. And now if you calculate, you know, 15 times 3, right? That's basically 45 nucleotides, right? So you do have proteins in the human genome that have 15 fingers and that have a potential basically to recognize a 45 base pair long piece of DNA, right? And those obviously don't, you know, belong to a family that you've characterized before. So those are hard to analyze in terms of specificity. And the other thing is that if you have a zinc finger that binds to a 45 base pair long motif, chances are that it's probably going to bind to one or two places in the genome, right? And therefore, you know, it's even harder to study, you know, using traditional approaches. And there are actually many of them. By and large, you know, most of them are actually not very well characterized. But you know, overall, I would say, you know, for most of these large families of transcription factors, we pretty much know what the specificity of the factors is like. Now, what we don't know is, you know, how they work together. You know, there are many ways in which factors can work together. You know, assemble in protein complexes. You know, and work, you know, cooperatively. You know, kind of bind the DNA cooperatively. And this is a big unknown. This is something that, you know, will take some work to unravel.
Just one more remark and following up on the previous question is that if you find a motif de novo, right, with fire or another algorithm, then you find a match to a weight matrix for some transcription factor. It doesn't mean that that transcription factor is the factor that's represented, that's controlling the expression, because what Olivier was saying, it's very important to say, to keep track of the direction. Like for a large number of transcription factors now there's a motif, but then there's large sets of transcription factors that have a very similar motif. So it's not an invertible relationship, right? If you have the motif, it's hard to uniquely implicate a specific factor. And then again, as Olivier was saying, you could hope to implicate the family of the transcription factor, right?
Yeah, and that's in fact what's happening here. So you see this guy ELK1, so that's an ETS family transcription factor. ELK1 is, you know, this ETS family transcription factor. It belongs to a family, and they'll pretty much all bind the same sequence here. So when we do the comparison between this motif here and the database of motifs, this ELK1 motif here in the database comes up first. But if you were to look at the second best, in fact the third best or fourth best and fifth best, they would all be members of the same family, right? And this one won by just a tiny bit, right? So, you know, we are definitely very careful when it comes to interpretation. We know that this motif belongs to a family, and, you know, we'd like to think that this is, you know, the best match is always, you know, the good match. But because there's always noise, you know, in the data, because the databases are not perfect, you know, they come from, you know, in vitro experiments that have their own biases, and this association is not very reliable, at least when it comes to the family member that's shown here. Maybe that's the second guy in the list. That's actually the real factor that binds to this motif in this particular condition. And the other thing is that, you know, things don't have to be expressed. You have a family of transcription factors. You know, some of them are expressed in some cell types, some of them are not, right? So maybe this guy is actually not expressed in this particular cell type in which we did the experiment. And it's the second best that would actually bind to this motif. So there are all these caveats that do apply. But it's just nice to have a sense of what kind of family of factors bind to this sequence here. It's useful. Okay.
So when you get a list of like potential cofactors from this motif discovery, and if you want to find like the top interest like cofactor, do you consider like the UI and MI and length of the motifs? And how would you, what would you suggest to consider the parameters?
So you mean, do you talk about changing parameters or what?
Like what, how, if I want to choose like one or two of the top motifs that most likely to be real.
Yeah. Well, you definitely have enrichment scores, for all of these factors. This is shown here, and then here. So you would tend to choose from the top of the list in terms of enrichment scores. The ones, but obviously, the ones that have a name are, easier to work with. This guy, good luck doing much with this, just because, you have no idea what to start. You don't know what binds to it. You have no clue what binds to it. At least here, you have a clue what binds to it. It may not be this factor, but at least, it's a factor that belongs to this family, most likely. So it helps a lot, in doing additional experiments. You could knock down this factor and see what happens to the binding of the factor that you did the experiment for. You could do a bunch of analysis based on this.
Going back to the difficult problem of the one that doesn't have a motif.
Yeah.
So that is potentially of great interest, right? Because you might.
It is, absolutely, yeah, sure.
So how would you, what are the best approaches to tackle this problem?
So, so basically what you want to do is to pull down the factor that binds to this, so you can make, you can make oligos, but you can make DNA sequences that have multiple copies of the same motif, right? You can put them on, beads, for example, right? And you can use that as a way to pull down whatever binds to the oligo, right? You use set extracts, right? You pull down the factors that bind to the oligo that has multiple copies of the same motif. You can sequence proteins using mass spec, for example, and that can give you an identity, something about the identity of the. It's just, these are difficult experiments because, the pull down is usually dirty. You get, you pull down a lot of things, and these are not easy experiments. But, these are things that people do for sure. Yeah.
In your case, you have, there seem to be only 3 positions that are important very important for that motif. So.
You mean this guy here? Yeah. I mean, I wouldn't say that. I mean, these do contain information. They do contain less information, but.
So you would I would expect without knowing so much how it works that you probably are not very specific in putting down stuff.
Yeah. Well. Yeah. I mean. Yeah. It depends on the protein that binds to it. It depends on the structures. The kind of bonds that it can form with each of these sequences. That's not necessarily the case. I mean, but yeah.
Are there separate public repositories for ChIP-seq data, or where do people typically?
So there are not yet, as far as I can tell. I mean, the ENCODE data is a great resource of ChIP-seq data. So we've done ChIP-seq for it. Actually, Mark Gerstein, the speaker today, will probably tell you more about this. But people have done, as part of the ENCODE project, we've done ChIP-seq for many factors across many cell lines. They've called the peaks, and they make that information available. It's available in the genome browser, which is nice, but it's also available for download. And you can, these are peaks, these are bed files basically, where you have chromosomes start and end, and you can do correlations between the ENCODE data and your peaks, for example. Using some of the packages, the tools that we've teaching you and will continue to teach you. It's just geoeng, overlaps between intervals and so on. Matt told you about the find overlap function in R, which you can use, for finding the overlap between an ENCODE set of peaks and your own peaks. And if the overlap is strong, it tells you that there may be some kind of shared biology between two sets of peaks and two factors.
So is there a way to compare different ChIP-seq data sets that are generated from different labs instead of just saying these peaks have the same peaks? Can you quantitatively measure?
Yeah, so you can definitely, this is basically related to the question we just discussed. You can download peaks, from ENCODE, right? And you can correlate them with your own set of peaks. Now, obviously, you have to be careful because, if you call peaks using your own ChIP-seq peak caller, and if, the ENCODE peaks are coming from a different peak caller, right? You're going to see differences just due to the peak caller algorithms that are different. So it's probably best to get the data from other groups of the ENCODE, call peaks using the same approach as the one that you use to call peaks using your own data set, and then do the merging, do the overlap between the peaks. Obviously, when it comes to peak overlap, you want to be careful, right? If you have 100 million peaks in your sample, and if you look at the overlaps between two sets of 100 million peaks, the overlap between the two sets of peaks is going to be gigantic, right? So you do want to shuffle, to randomize, peaks so that you can get a sense of what you expect in terms of overlap. The hypergeometric distribution here is a bit harder to apply to the situation because we're talking about genomic windows, that you can position randomly. But there are ways to shuffle peaks and to get a sense of the expectation in terms of number of peaks in an overlap operation just by shuffling the location of peaks. Yeah, okay, sounds good.
I think we should break now because we have a guest lecture at 4. Your tutorial is tomorrow.
Yeah, so we actually, it's up to you. So we have a small tutorial that we thought about doing today. It probably will be 10 minutes, 15 minutes. It's related to what we discussed. We can do it tomorrow if you think that's better.
Or tonight after.
Well, Matt is going to be leaving after later on. So you think we can do it now or not? We can do it tomorrow, relatively.
I think tomorrow is